Welcome to the Cannabis, Growth, Manufacturing, and Testing Learning Module, Section 2, the Cannabis Growth Process. The cannabis plant can be propagated through either seeds or clones. Seed versus clone selection is typically based upon the desired cultivar of the plant and the growing environment. Growing cannabis from seed results in a portion of the crop being male, unless the seeds have been genetically engineered to produce only females. Cloning, however, allows for the selection of female-only plants. Cultivars can also be crossbred to produce genetically superior plants. An example of this is a hybrid known as skunk, which is reportedly 75% sativa and 25% indica, and was bred to combine the high THC concentration of cannabis sativa with the growth and yield of cannabis indica. Typically, only cuttings of female plants are used for propagation of cannabis. Removal of male plants prevents the fertilization of female plants, which allows female plants to produce more flower with increased concentrations of resin and therefore increased levels of cannabinoids. Cannabis plants selected for growth should be free of pests and disease and of a healthy green color. Plants with a high concentration of yellow coloring should be removed, as this typically indicates a lack of chlorophyll, and growth of the plant will be less robust. In properly regulated cultivation, all propagation material, whether it be seed or clone, should be identified to genus, species, variety, and chemotype. Cannabis plants typically prefer neutral to alkaline clay and loamy soils with a good water-holding capacity that is not subject to waterlogging. The optimal soil pH for cannabis is 6.5 to 7.2. For hydroponic growth, nutrient solution is best at a pH of 5.2 to 5.8. Fertilizers should be used in a way that reduces leaching as much as possible, and if manure is used, it should be thoroughly composted and must be devoid of human feces. Growing medium should also be free of contaminants such as heavy metals, pesticides, and waste products such as sludge, not necessary for appropriate growing. This is due to the fact that cannabis is a bioaccumulator, meaning it will absorb these contaminants from the growth medium if they are present. Cannabis plants require controlled irrigation, which should only be applied based upon the direct needs of the plant. Overwatering can lead to rot or slowed growth and development of cannabis. Again, water should contain as few contaminants as possible, such as fecal contamination, heavy metals, pesticides, or other toxicologically hazardous substances. As the cannabis plant grows, exposure to short day-length growth causes the plants to change from vegetative growth to reproductive growth when male and female plants can be identified. Identification of plant sex during vegetative growth is difficult, although female plants tend to be stockier and flower later than males. Occasionally, a few flowers are produced in lower leaf axils during the vegetative phase, which allows for earlier sex identification of the plant. Typically, early, fully formed pre-flowers are used to determine plant sex. Flowering occurs as early as 4 weeks and is dependent on the plant cultivar and environmental conditions of the plant. Male plants are generally removed as they produce relatively low levels of cannabinoids. Additionally, pollination of a female plant by a male plant will significantly reduce the amount of cannabinoids produced in the female flower. However, some male plants are tested for their cannabinoid profile and used in breeding new cultivars with specific cannabinoid ratios. In the Northern Hemisphere, outdoor cultivation typically begins in late March to early April, depending on the environment. Full maturity of the cannabis plant when grown from seed is reached by 6 to 8 months, dependent on the varietal. Some auto-flowering strains mature from seed to harvest in about 75 days. The THCA content increases as the plant matures, reaching maximum levels at the full budding stage and for 2 to 3 weeks after, then declining with the onset of senescence. Outcomes are widely variable with outdoor cultivation due to uncontrollable environmental conditions. In several countries and U.S. states, medical cannabis products can only be grown indoors, where a controlled environment is possible. Indoor cannabis cultivation can occur in a variety of locations. The primary advantage to indoor growth is that it allows for the control of environmental conditions that can influence cannabinoid profiles, which will be discussed on the following slides. The disadvantages of indoor cultivation include pest control due to a lack of natural predators, soil composition because the nutrient content in purchased commercial soils can be varied and lead to deficient or excess nutrients, and artificial lighting, which must be carefully controlled to avoid burning or wilting of the cannabis plant. Cannabis requires high photosynthetic photon flux density for proper growth. Typically, fluorescent bulbs are used for juvenile cuttings, and metal halide or high-pressure sodium bulbs are used for established plants. Photo periods of 18 hours are optimum for vegetative growth, followed by periods of 12 hours to initiate flowering growth. For humidity, vegetative cuttings require regular water spray on the leaves to maintain a high humidity until the plants have rooted. Juvenile plants require high humidity of about 75%, and active vegetative flowering stages require 55 to 60% humidity. The optimal temperature for most cannabis strains is 77 to 86 degrees Fahrenheit. Often, indoor cultivators will use double levels of ambient carbon dioxide in order to stimulate photosynthesis and the efficient use of water in cannabis plants. This results in increased growth of the plant. The optimal amount and frequency of irrigation varies with varietal and growth stage, but typically, seedling and vegetative plants should be kept evenly moistened, while the top layer of soil should be allowed to dry in established plants. Electric fans are often used in indoor cultivation to help deter the growth of mold and facilitate more efficient gas and water vapor exchange within plant leaves. The process for growing cannabis indoor from seed is as follows. The seeds are planted in moist, aerated soil. Germination occurs within 4 to 15 days. This can be enhanced with heating mats under seedling pots. A photo period of 18 hours with cool fluorescent lights should be used during this stage. When roots are established, plants are transferred to a larger pot and exposed to full-spectrum lights. Once vegetative growth is established, the photo period is reduced to 12 hours to initiate flowering of the plant. Flowers should emerge within 2 weeks. When cannabis plants are propagated through clones, cuttings are taken from the lower branches of select female plants known as mother or mom plants and placed into a growth medium. Cuttings should be 6 to 10 centimeters long and contain at least 2 nodal segments. The growth medium can be soil or hydroponic. Typically, cuts are made at a 45-degree angle. The cut ends are then dipped in distilled water to avoid any air bubble formation in the stem, dipped into rooting hormone, and then placed in pots of growth medium. The plant should then be covered with growth medium up to the bottommost node of the cutting. Rooting occurs in 2 to 3 weeks. After 6 weeks, plants are transported into larger pots, and the same growth process is followed as for seed propagation from this point forward. Pests, bacteria, and fungus can all be associated with both indoor and outdoor cultivation. Outdoor plants are relatively pest-resistant, and commercial pesticides are often not needed. However, small animals can eat emerging greenery, and runoff from other crops may contaminate groundwater or soil. Fertilizer choice has a significant impact with outdoor cultivation of cannabis. There have been outbreaks of hepatitis A reported with use of cannabis from Mexico, where human excrement was used as fertilizer. In indoor cultivation, a lack of natural predators can make pests an issue, particularly spider mites, thrips, aphids, and white flies. Natural predators are often used to mitigate these pests. Any chemical pesticides used must be safe for human consumption. In indoor cultivation, fungal root rot can easily occur if the growing medium is too wet or hot or with an excessive use of nitrogen-rich nutrients. For both indoor and outdoor crops, powdery mildew is a significant issue and can be caused by excessive humidity, excessive shade, and a lack of air circulation. The application of any treatment for these issues must be carefully considered and only applied if there is sufficient time for the agent to be cleared from the plant prior to harvest, as many commercial pesticides can pose a health risk if consumed.